Hi, our today's topic is DNA and the helical structure of the DNA. The structure of the DNA is referred to as a double helix as it resembles a twisted staircase. A molecule of the DNA comprises two strands wound around each other as a though a twisted ladder where each strand comprises a backbone comprising alternating group of phosphate and sugar groups. The nitrogenous bases are centrally placed, holding together these two strands. One of the four bases is attached to each of the sugar. A is adenine, G is guanine, C is cysteine, or T is thymine. Both strands are united with the help of bonds between the bases. Adenine forms a base pairing with the thymine, whereas the cytosine pairs with guanine. This pairing is referred to as the base complementary rule since the DNA strands are complementary to one another. Thus, when the sequence of the strand is TTGGCCAA, then the complementary strand should have the AACC, GG, and TT as its sequence. As a result of this base pairing, DNA strands are antiparallel, complementary to one another, and running in the opposite direction. This is to stay. The 5 carbon end of one of the strands faces 3 carbon end of its complementing strand. This orientation that runs antiparallel is crucial for replication of DNA and several nucleic acid interaction. Each DNA strand is a double helix is linear and long, comprising smaller units known as nucleotides, which is in turn from a chain. The chemical bond referred to as the sugar phosphate backbones are formed by the phosphate and sugar molecules. The WHO proposed this structure. The James Watson and Francis Crick pioneered in 1953 to put forward the molecular structure of DNA, which they termed the double helix in a journal. Now the DNA replication. Each strand of the DNA copies during replication of the DNA leading to the formation of a daughter DNA double helix. It contains one parental DNA strand and one newly synthesized DNA strand. This is a possibility at this juncture for any change in the sequence of the nitrogen base to take place in a short mutation to occur. In most cases, the DNA proactively takes charge when mutation occurs and is thereby able to fix itself and gain back its original base to the sequence. On the other hand, when repair is not seemingly a possibility, new protein takes birth. To summarize, the unique structure of the DNA allows molecules to copy itself while the cell division process is on. Wherever a cell is ready for division, the DNA helix breaks into two and identifies itself as two individual strands. The single strands hence act as a template to build two double-stranded DNA molecules, each of which is a replica of original DNA molecules. While this process is on, adenine bases are added where there is a thymine, and similar is the case with C and G as aforementioned. Consequently, these bases now have partners once again. During the formation of proteins, this double helix unwind enabling a single strand of the DNA to act as a template which is in turn transcribed into the mRNA. The mRNA is a molecule conveying crucial instruction to the protein greatening factors of the cell. This was a brief of the double helical structure of the DNA. Next is DNA polymerase are a group of enzymes required for DNA synthesis. Arthur Kornberg purified and characterized the DNA polymerase from E. coli for the first time. It is a single chain polypeptide now known as a DNA polymerase 1. Scientists have now found 5 DNA polymerase in E. coli. DNA polymerase definition is DNA polymerase are a group of enzymes that catalyze the synthesis of DNA during replication. The main function of the DNA polymerase is to duplicate the DNA content of the cell during cell division. They do the by adding nucleotides at the 3OH group of the grouping DNA strand. So this is information related to the DNA replication. Thank you.